Somebody squawk if you can't. So I'm going to talk about the dissolved oxygen monitoring that we're doing. This is collaborative work and definitely heavily reliant on these collaborations, both with the department level GIS team, with the Mass Lobstermen's Foundation, and with the Center for Coastal Studies and Woods Hole Oceanographic Institution. So in order to sort of set the stage here, we have to go back to last fall, when in mid to late September, we started receiving phone calls from lobstermen in Cape Cod Bay that they were hauling up anywhere from dozens to hundreds of dead lobsters in their traps. They were also seeing dead crabs and finfish at the same time. And the time period was pretty concentrated right around September 20th to the 24th or so. We also received a report from a dragger who worked in the area of much higher incidence of dead scallops in his catch than would be, remotely normal. And again, these were all focused in sort of the southwestern corner of Cape Cod Bay, roughly from east of the canal over to sort of the Barnstable Harbor entrance area. And it seemed to be isolated within the depth range of about 30 to 80 feet. So we were taken totally by surprise here, and we really didn't have a good idea of what could be going on. So we really scrambled some staff to try to start learning about this and trying to figure out, what could be happening here. So, we looked into getting folks on the water to take some water quality readings. We had a lot of assistance from Center for Coastal Studies with this. We sent some of our biologists out aboard the commercial boats working in the area so that we could directly observe the catch as they were seeing it. We also put some divers in the water so that we could do direct observations of the bottom. We took a lot of time and spent a lot of effort looking into the location and timing of some of the mosquito spray events, mosquito control programs. We also took some of the dead lobsters and fish and sent them away for pathology analysis. And we spent a lot of time collecting data on the oceanographic and weather patterns in the area time. As we were going through this, essentially the water quality results really became sort of the smoking gun here. And what we were seeing is evidence for some pretty severe hypoxia. Hypoxia just means that there's not enough oxygen in the water. And the readings we were getting back in the area were extremely low. So we're talking about dissolved oxygen here and levels less than 1 milligram per liter in our samples. These levels are definitely lethal to lobsters after about 48 hours. So it became pretty obvious of what the major problem was, is a severe hypoxia. And when we looked at all of the data we collected from last fall, we can kind of map it out. And, a relatively surprisingly large portion of the southern portion of Cape Cod Bay is experiencing or was experiencing this low dissolved oxygen. And if you look at the map here, anything that's shaded in the yellow is what we would consider to be a low dissolved oxygen. And this is less than 6. The orange here is what we would consider to be hypoxic, less than 4 milligrams per liter. And in the red here is what we would consider to be severely hypoxic, or, if we can euphemism, this is the bad area, this is the dead zone. And that's less than 2 milligrams per liter. So just real quickly, how does hypoxia happen or form? Essentially, it's the result of large inputs of nutrients coming into the water. Those nutrients essentially form the basis to allow a huge increase in organic material, so algal blooms. Then when that algae dies, it sinks to the bottom and decomposes. The process of decomposition uses oxygen from those bottom waters. And what happens is when you have a stratified water column or a very layered water column, the surface waters are prevented from mixing down and replenishing that oxygen in those bottom waters. So if you can't get that mixing over time, the decomposition sucks up all the dissolved oxygen, and the result is that you get this hypoxic condition. Generally, mobile animals like lobsters, crabs, fish can move away from hypoxia to avoid those conditions, but anything that's less mobile or obviously stuck in traps cannot move, and it will kill them. One of the most sort of infamous dead zones or hypoxic areas in the US is this in the Gulf of Mexico. We've probably all heard of this. This is the result of massive amounts of nutrients that come down through the Mississippi are released into the Gulf of Mexico and cause these huge dead zone. This is just the extent of the 2019 version of the dead zone. So our concern here was whether or not Cape Cod Bay could be going this route. So in doing all of the homework that we did last year to try to understand this, we discovered that it actually happens annually. So seasonal declines of dissolved oxygen in Cape Cod Bay is a relatively normal event. What happens is that for most of the year, Cape Cod Bay is essentially downstream or down current of this heavily populated coastline, which means all of the nutrient inputs come downstream into Cape Cod Bay. But into the summer and fall, there's some changes in water circulation patterns that tend to then isolate Cape Cod Bay from the rest of Massachusetts Bay. And if you look at the maps here on the bottom left, shows sort of what the spring water flow conditions look like. And you can see there's a very strong current that brings nutrients down into Cape Cod Bay in the spring. But then in the summer, what happens is the water circulation patterns change, and instead of coming down into Cape Cod Bay, the water currents are sort of deflected off to the east and end up going out and around the backside. And what happens within Cape Cod Bay here is you get these little sort of gyres that set up that are sort of relatively isolated. And this one down here that sets up in the southwestern corner just happens to coincide with the area where we saw all of the hypoxic conditions. The other characteristic of Cape Cod Bay that sort of sets the stage for this sort of declining oxygen is a very strong thermal stratification that tends to set up in the summer and then persist into the fall. And again, stratification is just layering of the water column. And the temperature data here in this graph shows, so this is the surface. And at the surface, we've got 18 degrees centigrade, which is about, I think it's 64 Fahrenheit. And it's very warm all the way down to about 60 feet in depth. And all of a sudden, there's this drastic change. So down on the bottom, you're looking at maybe 8 degrees centigrade. So it's a huge temperature change over a relatively narrow depth range. And so this is a very strong thermal stratification. And this kind of setup is what prevents these warm surface waters from mixing down to the bottom. So the question really is, are we looking at the southern portion of Cape Cod Bay developing into sort of a new annual dead zone? As we all know, the Gulf of Maine is warming more rapidly than many other portions of the world's oceans. Warmer waters tend to increase the potential for hypoxic conditions to develop. Warmer waters hold less oxygen. They tend to further stratify the water column. They increase the biological productivity, so more stuff that's going to die off eventually and decompose. And then it also increases the sediment oxygen demand. So there's more demand for the oxygen to break down all that material. So in order to really answer this question as to the future of Cape Cod Bay, we really need a better understanding of what's driving these processes and try to figure out whether or not 2019 was an anomaly, whether it was going to stand out or if it was going to become more normal. And just to give a little bit of information here. So the graphics that just popped up on the right here, this is chlorophyll A data, and chlorophyll A can be used as a proxy for algal biomass. If you look at this top, this is September average chlorophyll A over the time period of 2010 to 2018. When you look at the bottom, this is the September values in 2019, and you can see the huge difference that makes 2019 look really different from this time series average. Suggesting that in 2019, there was an awful lot of algal biomass in the system here. So in order to really understand this and whether or not, what we saw in 2019 is sort of going to become a new normal, we really needed to develop some new monitoring efforts to try to get a handle on this and to try to get enough data to inform us. The first of these new monitoring efforts is the creation of the Cape Cod Bay Study Fleet. So in 2019, when all this happened, the fishermen in the area expressed very strong interest in getting involved and helping us to monitor the situation moving forward. And the Lobster Foundation of Massachusetts was able to secure funding through a state grant program this spring, that sort of set up the this study fleet. So the funding was used to purchase data loggers and data deck boxes to outfit 5 vessels as sort of monitoring platforms. So the data logger, which is this black thing here, records dissolved oxygen and temperature. And then the data deck box here, each boat has one of these. And the logger is attached to the traps. They go down with the traps, record these dissolved oxygen and temperature every 15 minutes. Then when the traps are hauled, the logger just automatically through a Bluetooth connection detects the data box, downloads the data, the traps are set back, the logger goes back in the water, and then the deck box here sends the data through a cell phone signal to a server on land. So it's a super efficient system, and it lets us get a lot of information in place, which is really nice. So the fleet was able to deploy these loggers this July. And this chart here just gives you a good idea of sort of where all of the loggers were deployed. These are through the end of September is what these locations show. So you can see we've got pretty good coverage in this southwest corner of the bay here. And we've also got some coverage up here in the northeastern corner. So the other portion of this work is collaborative work with the Center for Coastal Studies, a couple of researchers at the Woods Hole Oceanographic Institution, and again with the department level GIS team. And we were able to secure additional money to work on this through the National Sea Grant American Lobster Research Initiative, with the goals being to increase our ability to detect and hopefully predict the potential onset of hypoxia, and then also to figure out a way of efficiently alerting the commercial lobster fleet and other stakeholders that the conditions are changing so that we can protect the fishery resources. So this work focuses on, again, water quality data collection, but also some data mining. So looking into historical data sets to try to figure out what things looked like in the past. Oceanographic modeling, some near real-time mapping. So we're trying to develop some fancy maps that let us look at the data on a near real-time basis. And then also integrating some catch monitoring so that we can try to understand how the lobsters are reacting to these changing environmental conditions. So Center for Coastal Studies already has an existing monitoring program in Cape Cod Bay. They monitor 3 stations specifically for MWRA, and then they have an additional 9 stations, and they do the monitoring work monthly. So this work is adding an additional roughly 6 stations to their monthly sampling. And then what we're going to do is look at, so we will monitor the data coming in from the study fleet. And as soon as we start to see the conditions sort of declining, that dissolved oxygen coming down, that will be the signal to initiate some of these high resolution transect sampling. So the CCS will focus sort of on the southern portion here with these north-south transects for high resolution sampling, with the idea being to get a better idea of sort of the spatial extent of these hypoxic conditions. Center for Coastal Studies sampling is a little different from the study fleet. The study fleet is taking one sort of point rating right on the bottom. The Center for Coastal Studies instrumentation allows them to take samples throughout the entire water column, so from the surface all the way down. And the data they'll collect is dissolved oxygen, temperature, chlorophyll, and a few other things. So don't strain your eyes trying to figure out what all this says. I just want to illustrate that in 2020, we were very successful and collected a whole lot of data. I'm not going to go through this in detail, but I do want to point out some sort of important things that we learned with all of these new data. So this kind of gives you an idea of what we're trying to work towards with the mapping. What you can see here is the spatial extent of the study fleet and the color coding of the values that are coming back. So I blew the legend up here real big, just again to get everybody on the right page. Essentially, if it's green or blue, it's normal. Yellow is low, orange is hypoxic, and red is really bad. And essentially each one of these data points is one of those 15-minute readings. Don't pay any attention to the size of these points. The size changes just so that we can visualize multiple points stacked on top of each other. But what I really want you to look at here is sort of the spatial extent of the data that we're getting in, and then a couple of trends. So firstly, we are seeing the yellow values, which indicates low dissolved oxygen, pretty widespread throughout the August through October time period. And so a lot of the loggers in the southwest region were detecting hypoxia and severe hypoxia. So down here in the September map, this is the southwestern portion where you can see a lot of the orange and red dots popping up. One caveat here, which is why I'm calling this pre-QC data. So we have not yet filtered out all of the sort of the noise and the erroneous readings here. So for example, this logger up here in Provincetown is coming back with a severe reading. We think that this is a erroneous reading. Probably something like a piece of algae was blocking that sensor briefly, and it came back with a very low reading, but we don't think that the Provincetown area actually experienced any hypoxic conditions. You can see it happened again once in September, but all of the other readings were fine. And the same thing happened over here in the Manamet area. We think that this is an erroneous reading. So we have some improvements we need to make to this system. What we're going to try to get to is probably, instead of trying to visualize every single reading, we'll probably try to look at it on like a 6-hour average. We think that'll be more informative. So if you look at some of the study fleet data, I've selected the Sagamore region for the loggers here. And if you're not familiar with Sagamore, the map down here on the bottom, here's the Cape Cod Canal at the very bottom of the screen. So the Sagamore region is immediately north of the Cape Cod Canal. And this data set is actually nice and clean. It's one of the easiest ones to deal with in terms of there's not a lot of errors in here. So the dissolved oxygen is shown in blue, temperature is shown in orange here, and we're just going to go through the dissolved oxygen trends real quickly here, because there's some pretty good signals here. So the first thing we see is starting from about mid-August through the first half of September, all 5 loggers are showing a really strong signal of declining dissolved oxygen, reaching down into the levels, particularly in this bottom one down here, of hypoxic conditions. So after that, we had a brief improvement period in about the middle of September or so, where we saw a pretty quick improvement in dissolved oxygen. But then we saw another decline. This took place sort of early October area before it did improve again. And then sort of in mid to late October, we get this weird little blip where 4 of the 5 loggers picked up this decline signal. So we are seeing a lot of dynamics in what the dissolved oxygen is doing. So with the Center for Coastal Studies survey results, the decline that we saw in the fleet loggers in August is what initiated the transect sampling. So we saw it was coming down and we knew it was time to get out there. Center for Coastal Studies did these surveys roughly weekly, and depending on which boat they had and what the weather conditions were, we're able to accomplish more or less depending on the day. So the first day they got out, they got 1 transect done. This is August 31st. The color coding is the same as what you've been looking at. And you can see here that the severely hypoxic waters is in this sort of 60 to 75 foot depth range here. Roughly a week later, September 3rd, you can see the severely hypoxic mass has shifted to the south into much shallower waters. So we're in about 30 to 50 feet here. A week later, it's still in that shallow southernmost portion of the bay. And then the week of on September 16th, now we see the mass has shifted north out here into deeper waters. So we're looking at it in 70 to 90 feet of water here. And the little red box over here, I put this in because this is actually the only reports we got this year of dead lobsters occurred in this little red box area. During the time period, the gear was fishing from the 16th to the 24th. So we know that something bad happened in the area of that little red box. But the next time the Center for Coastal Studies got out on the 24th, we saw a massive improvement throughout the area. So all of these stations are now reading with normal oxygen levels. But again, you can see the little red box here this is the region where we had the dead lobster reports. So at some point in time, that hypoxic water mass must have shifted over this region here. So with seeing all of this, the data coming from the Center for Coastal Studies and the variation that we're seeing with the study fleet data, we know that this dissolved oxygen, this hypoxic water mass is moving around. And the best way that I've found to sort of conceptualize this and think about it is I called it the blob. So the blob, I went with this because it is kind of a pretty cohesive water mass that it's best to think about it in 3 dimensions. So it's this cohesive mass. It's generally located very close to the bottom. And it does shift around. So we think that the shifting is being driven by upwelling and downwelling events that are wind driven, so that when we get a strong period of northerlies, the northerlies push the surface waters towards the southern shoreline, which creates a downwelling condition along that shoreline. The downwelling then pushes the blob out away from shore. Whereas if we have southerly winds, the southerlies are pushing the surface water away from the shoreline, which sort of allows the bottom water to come in and produces an upwelling. And that's what brings the blob in closer to the shoreline. So if we go back again and have a look at the Center for Coastal Studies transects, you can see, again, the red dots here, the blob is inshore. And we're going to take a closer look at transects 4 and 5 here, which I have labeled in the chart here. And again, with the Center for Coastal Studies data, what they're able to do is take an entire water column profile. So that's going to allow us to look at this in 3 dimensions. So if we look at transect line 4 here with their water column profile, so think of this as we just took a slice and we're looking at the water from the surface down to the depth. So on these charts here, the top one is temperature, the bottom one is dissolved oxygen. This is essentially the distance from shoreline. So this is, this is shore, this is moving north away from the shoreline. And then this is the depth on the vertical axis here. So the first thing you can really see here is with the temperature, you can see that really strong thermal stratification, extremely warm surface waters, and much, much cooler bottom waters. And then with the dissolved oxygen, what you're looking at here is anything from like the green into these warmer colors. This is the severely hypoxic range. And so you can really see the blob here in its, height dimensions, so that you can see the minimum is right on the bottom, and it's about 1 kilometer offshore here. And the top of the blob, so the top of the yellowy green here is about 6.5 feet off the bottom. And it extends it out another kilometer or another half a mile offshore. So the whole extent of this thing, both vertically and then offshore, is not small. If we shift one transect line over to the east, you can see it looks a little bit different over here. You still see the strong thermal, the strong stratification in the thermal signal here. But what we look at here, now the blob looks like it's up off the bottom a little bit here, and it looks a little bit smaller here. It doesn't quite extend as far in the north-south direction as it did in the western transect. So you really get an idea of sort of how this thing looks in 3 dimensions when you use the water column data along with the transects. If we go look again at the September 16th, where it's now been pushed offshore, you can see from the vertical profile, this warm surface water has now been mixed down near the shoreline. So that warm water is now down at the bottom close to shore. And you can see here, the blob has been pushed way offshore. So we're 6 kilometers from the shoreline here now, where that DO minimum is. And this is the result of northerlies and that downwelling I talked about. What we think happened here is the September 18th through the 23rd or so, there was a period of pretty strong north and northeasterly winds. This is the buoy data of wave heights. And you can see we had this sort of extended period of like 3 to 6 foot seas for several days. We think that that's what caused the downwelling and pushed the blob offshore. And when we go back to the September 24th, after that wind period, now you can see, okay, it's been, for the most part, it's been mixed out here. But again, we had this dead loss here. And what we think happened is during this period of the strong northerlies and the high seas, the first thing it did was push the blob offshore. and it probably moved through this area and unfortunately got into a couple of guys' gear here. And then finally, because of the high seas, the surface waters were mixed down and the oxygen was replenished, but it took a couple of days of that before it finally seems to have broken up and dispersed. So what we've really learned from our 2020 work is that we are seeing a relatively large portion of southern Cape Cod Bay experiencing low dissolved oxygen. And it seems to happen starting in mid-August and through September, and then definitely extending into October in some instances. So now we have 2 years of 2019 and in 2020 where we have detected actually hypoxic conditions, so conditions that are not good for bottom animals to breathe. The declining DO results in the formation of this three-dimensional hypoxic water mass, or what I call the blob. The depth of the pycnocline, which is essentially water density, and it sort of comes into play with the temperature also, but that density change is likely what's controlling its height and keeping it pushed down towards the bottom. So all of the dense water is concentrated on the bottom. And that the blob moves. So this movement seems to be driven by wind events that cause upwelling and downwelling conditions. We do think that there is a period of high seas are required to mix the water column and replenish the oxygen on the bottom. And we should maybe expect that when we get these strong northerlies, it's going to help, but the first thing it's going to do is move the blob into deeper waters. So, you know, when we see the winds pick up and when we see the northerlies and the sea state pick up, that's probably a good thing for getting rid of this. But it doesn't mean that somebody's not still going to be affected by it until it moves, until it gets totally mixed out. We think that 2020 was a little bit less severe of an event than 2019. We certainly had fewer reports of dead lobsters or dead anything in the traps. We only had one of the fleet's data loggers record a prolonged period of severe hypoxia. So only one logger had about 48 hours worth of that severely hypoxic death zone kind of readings. So that's, I guess, good. We do think that it appeared, at least to me, to be a little bit of a larger east-west extent this year than any of the data we had from 2019 indicated. So for example, we didn't have any reports or get any readings near the mouth of the canal or sort of to the north and west of the canal of low dissolved oxygen, but we definitely did this year. So whether that was a timing thing on when we were out there, we certainly had a lot more data in 2020 than we did last year. And that does make it a little bit hard to compare. So these last 3 points are in italics because these are sort of my caveats. Because we had so much more data in 2020, it does make it a little bit hard to compare for 2019. We just didn't have as much data. We're still missing data sort of in the middle of Cape Cod Bay. So the mid portion here, we would like to target that to getting some additional readings in that area for next year. And then the study fleet does move their gear. So the guys reacting to the data that they're seeing recorded on the deck box might be allowing them to move their gear, and that might have actually helped alleviate some of the dead loss, which is what we hope will happen, but it does make it sometimes hard to interpret the data. So the next steps here, you know, we're thrilled with how this year went. The success of the study fleet has been phenomenal. The oceanographers at WHOI are very excited about the quantity and quality of data that the study fleet is producing. So this has been great. We are planning to continue the fleet next season. I'm hoping to expand it with maybe another 1 or 2 vessels, specifically to try to cover the central portion of the bay. The collaborators at WHOI will be working towards identifying what drives these declines in dissolved oxygen, trying to get at it in more detail and really better understanding of the process. And they'll be using the data from the fleet, from Center for Coastal Studies, and essentially anything else that they can dig up that is going to be relevant. DMF and then Fish and Game staff are going to continue the development of these mapping applications. You know, eventually we want to get to a web app that will display current and recent conditions that the general public can access and look at to see what's going on. And we're trying to develop a smartphone application that will allow the fishermen sort of while they're on the water to look at, all right, what are the conditions right where this gear is, or right where I want to put this trawl, to help them sort of make informed decisions about where to place the gear. And then we're going to add this catch log to the study fleet for next year. And what we'd like to ask them to do is to sort of fill out a log that will help us link what they're seeing in their catch to the environmental data that they're collecting, again, to try to understand how the lobsters are reacting to these changing conditions.